Climate change is reducing biodiversity in a lot of living organisms, and one of the most vulnerable animals is ectotherms since they regulate their body temperatures using external sources such as sunlight or shade. Therefore, it is crucial to understand how heat stress affects them. In this study, we're using Drosophila melanogaster, which is an ectotherm, to study the effect of high temperatures on development and fertilization. Two different genotypes were employed: temperature-sensitive wild type and temperature-insensitive TRPA1 knockout animals.
How does the absence of heat avoidance mechanisms impact the development success of Drosophila melanogaster? And is the fertilization process disrupted at high temperatures? We hypothesized that despite the increase in mating under heat stress, fertilization will not be affected in TRPA1 flies, which are unable to sense temperature.
To test this, virgin females were collected and allowed to mate on grape juice agar plates at either 20 or 30 degrees Celsius for 24 hours. We then counted the number of eggs the following day, the number of pupae a week later, and the number of adult Drosophila 2 weeks later. We found that wild-type flies had fewer embryos at 30 degrees, but temperature had no effect on TRPA1 flies. Temperature significantly affected some other developmental stages in both genotypes.
Since mating behaviors are the same in TRPA1 Drosophila, this suggests that while temperature may affect general development, it does not affect the process of fertilization and the release of the egg.
One major challenge with our study is the low power due to a low replicant number, resulting in some promising trends in this pilot data, but not always significant differences. Future studies would increase the replicant number and test other temperatures, including colder temperatures, to further characterize the effects of temperature on every developmental phase. If we are able to characterize this more, it will shed further light on the impact of climate change on ectotherms.